The overall goal of this procedure is to behaviorally evaluate visual system function in larval and adult zebrafish in order to quickly identify mutant larvae with visual system defects, or to compare visual system properties of wild-type and mutant or morpholino-treated animals. This is accomplished by first restraining body movements of the experimental animal. Larvae are embedded in methylcellulose, and adult fish are anesthetized and fitted into an immobilization device. Next, place the body-restrained animal in the VisioTracker in the center of a drum-shaped screen. Vertical black and white stripes are projected onto the inside of this screen using a digital light projector. Eye positions are then recorded and analyzed automatically by the VisioTracker software package, which also calculates eye velocities in real time. The final step is to process obtained data, relate eye velocities to the properties of the visual stimulus, and calculate average eye velocity and standard error for each experimental condition.
Though this method can provide insight into visual system properties of larval and adult zebrafish, it can also be applied to other model organisms, such as Medaka, Kupi, and virtually any other small teleost species.
I first had the idea for this method when we used the optokinetic response to screen for blind larvae and quickly got interested in larvae that are not completely blind but rather visually impaired. Such mutant larvae potentially affect aspects of visual processing.
The system includes an immobilizing device for small fish positioned under a high-quality video camera equipped with a zoom lens.